The Genome Challenge. Hello everyone, this is Dhruv of class 12th of Rahman Singh Senior Secondary Public School, Najafgarh, presenting the Genome Challenge. So first of all, let us discuss what is genome. Before discussing genome, we should know about gene. So a gene is a segment of DNA that contains the instructions for the production of biological molecules, usually called proteins. Humans have around 20,000 genes, all containing the information needed to build one or more proteins. Some genes determine physical characteristics such as the color of our eyes. Humans have 23 pairs of chromosomes, with one of each pair inherited from our father and the other from our mother. Now let's discuss what is biotechnology. So, biotechnology is a science that is based on biology. Even life form is made of DNA, which are the building blocks of every life form. By modifying these building blocks through biotechnology, we can make the food healthier. At the same time, resistant from bugs and pests. By manipulating these building blocks in another way, we can copy traits of an imported rice that has high threshold in water, then combine it with the local rice so that it can survive in the country. Biotechnology has a lot of advantages. So let's discuss its advantages. It helps to fight with malnutrition. It provides a lot of vitamins and minerals. It gives high yielding crops, and it may enrich in the economic and agriculture income. Now, what do we understand by the word GMO? So, GMO stands for genetically modified organisms. GMOs are living organisms whose genetic material is artificially manipulated. This creates combinations of plant, animal, bacteria, and virus genes that do not occur in nature. The technology enables us to increase our food production and make it more efficient. Modified food crops will be insect resistant, resistant to fungal infections, and also resistant to extreme weather conditions. They will grow faster, need fewer acres to grow, and certain more nutrient ingredients. GMOs can fight malnutrition and decreases carbon dioxide emissions, and also decreases water usage of green leaves. Now, what do we get from the restriction enzymes? So these are the molecular scissors that can cut DNA at specific locations. Today, there are more than 900 restriction enzymes isolated from about 230 bacterial strains. The restriction enzymes are divided into two classes, exonuclease and endonuclease. Both are present in bacteria, but they do not cut bacterial genome. So now one by one discuss both. What do we understand by endonucleases? So endonucleases are those enzymes which cut at the center of the DNA. For example, deoxyribonuclease 1, ecorib 1, BAMH1. The next is exonuclease. Exonuclease are those enzymes which cut DNA at the end. For example, snake venom, exonuclease 1 and exonuclease 2. Now let's discuss what is plasmid. Plasmids can be a good cloning vectors because they carry an origin of replication and are therefore able to replicate independently within a cell. Most plasmids used as vectors also encode some type of selectable marker such as the gene for resistance to amplicin. What if the gene of interest is already extracted? Then it is required amplification of DNA that is done by PCR. Now what is PCR? It stands for polymerase chain reaction. This is the technique that can produce over a million-fold amplification of target DNA within a few hours. It consists three steps: denaturation, annealing, and extension. So in denaturation, it takes place at the temperature of 95 degree Celsius. In this method, In this method, the DNA is separated, and the next method is annealing. This method hybridization takes place at the temperature of 55 to 65 degrees Celsius. And the last step is extension. In this method, the new DNA is synthesized. This step requires 72 degrees Celsius, and it takes place in the presence of a thermophilic bacterium, Thermus aquaticus, which is known as stack polymerase. So it requires 20 nucleotides. Here is the animation. You can see all the three steps. Now, what do you understand by the term host competent? So in this, there is first method is gene gun, which is takes place in only and only in plants. In this, cells are bombarded in the plant cells, and the DNA is then inserted with the help of gene gun, with the help of gold or tungsten filaments, and it is also known as biolistics. The next method is microinjection. In this method is useful for animals. In this method, the new DNA is directly injected into the cells of animals. The last method is heat shock method. In this method is only used for bacteria. In this method, the bacteria is heated